How's it going? I hope your answer to that question was fantastic because you'll need it for this. My name is Jackson Durker, and today we will be reviewing and discussing a piece of primary literature. This piece of literature is a research article detailing the findings of a research team in China. They are looking at how the extract triptolide, originating from a local Chinese herb, can induce GSDME-mediated pyroptosis in head and neck cancer by way of suppressing hexokinase-II found in those cells' mitochondria. With introductions out of the way, let's get into it. As I mentioned before, this study is focusing on a compound called triptolide, or TPL for short. TPL is extracted from a local herb called Trigium wilforti hookef. Hopefully that's how you pronounce it. The study is investigating the quality of which TPL can induce pyroptosis. No, I did not pronounce apoptosis incorrectly. This is actually a newer term that came about around 2015. Pyroptosis is the act of inducing programmed cell death, but through the means of pro-inflammatories. This differs with apoptosis because apoptosis is the overarching term used to define programmed cell death, so it incorporates all the ways that a cell is able to undergo cell death. So think of pyroptosis as a way to further narrow and describe the way apoptosis occurs to a cell. In regard to pyroptosis, this study is focusing on the gasdermins, or GSDM for short, family of proteins. Evidence provided throughout this presentation will hint at why they chose to focus on GSDME. A letter at the end of GSDM, like the E in GSDME, is just to signify a specific protein in the family. The GSDM family is known as pore-forming proteins. In essence, this just means that these proteins cause holes in a cell's lipid bilayer, which ultimately leads to cell death. Now, you're probably wondering what hexokinase-II, also called HK2, my apologies, for short, which you'll see in the paper. Well, since you're so curious, I will tell you. Hexokinase-II, like its isoform, hexokinase-1, also known as HK1 for short, allows for the processing of glucose in carcinoma cells, which is just a scientific way of saying cancer cells. Hexokinase-II is the main leader in kicking off rapid glycolysis. Because cancer cells divide so fast, they need lots of energy. So, unlike hexokinase-1, hexokinase-II can meet these rapid metabolic needs of carcinoma cells. They can meet the rapid metabolic need of carcinoma cells, which make it a good target for a study like this because they can target the cell's energy production. Throughout this study, they also looked at other characteristics to fully comprehend how this TPL inhibition mechanism can lead to pyroptosis. They looked at c-Myc, which promotes the expression of hexokinase-II. They looked at the cysteine glutamate antiporter, which was responsible for bringing cysteine into the cell and maintaining homeostatic conditions with reactive oxygen species, also known as ROS. The ROS was an important condition for them to look at because the accumulation of ROS can cause damage to DNA and ultimately lead to cell death. Throughout this presentation, though, we won't really be going into too much detail about that. Here, we have figure 1A, and this is what kicked off the whole study. This is a cell viability assay, where they looked at 3 different carcinoma cells, C666-1, HK1, and Fadu cells, as I call them. Not actually sure if that's how the authors intended it to be pronounced. In this figure, they exposed each of these cells to varying concentrations of triptolide, found that triptolide did reduce the viability of these cells. This effectively means that in some way triptolide was causing these cells to undergo cell death. Curious, isn't it? And we can also see down here on the x-axis at around 150 nanomolar is where they saw significant decreases in cell viability. Now, I know we just got started, but we're going to take a slight detour to the lands of polymerase chain reaction, more commonly referred to as PCR. We are taking this detour because the figures we'll be focusing on in this presentation were conducted using PCR or a variation of it. This will just serve to help in our understanding of this paper. So, ever since COVID came about, the term PCR has been thrown around a lot. But what even is PCR? PCR is the act of taking a target strand of DNA or multiple strands of DNA and amplifying those strands into hundreds of millions, billions, or however much DNA your heart desires so that it can be used for some scientific purpose. So it's all done by saying, with your sample that contains your target DNA. Is it? Oh, sorry. This is all done by starting with your sample that contains your target DNA. The sample will go through multiple buffers, which are often provided in kits by large pharma or research companies that will break the cell open, chuck out all the insides you don't want, and leave you with that cell's DNA. If we look at the picture that I've provided, this huge thing, we can get a pretty decent visual overview on how PCR works. Feel free to pause the video if you want some more time to look over that image. We'll be going into more detail in each part of that image in a moment. Alright, that was a pretty fast moment. When your DNA is done being purified, a small portion of that sample can be run through spectrophotometry. This is the act of passing light through the sample and determining how much of that light was absorbed and at what wavelength. Don't worry, a machine will do this for you so you do not have to manually try and do this yourself. When the process is complete, depending on software and such, it can pump out a spreadsheet that looks sort of like this. You can see that B, C, and D are all individual samples. If we look here at the bottom of each sample, we can see bolded numbers. This is the concentration of DNA that was in the sample in nanograms per microliter. This value will become important for calculating how much we'll need to go into our PCR reaction mixture. How much sample will need to go into the mixture, that is. Okay, this scary looking table, full of numbers and named columns, is an example of a PCR reaction mixture. This scary looking table full of numbers and named columns is an example of PCR reaction mixture calculations that I did during my time in undergrad. As you can see by what I put in the box at the top right of the slide, right here, in each PCR tube, you will need your desired primers, some type of Taq polymerase for adding nucleotides to the DNA, some free-floating, well, not some type, it's going to be Taq polymerase, for adding nucleotides to the DNA, some free-floating nucleotides for elongation, and likely some water. In this example, red Taq, which we can see right here, includes the Taq polymerase and the free-floating nucleotides, which was quite convenient. For our reactions, you'll notice that we included our template, the red Taq, a forward primer, and the corresponding reverse primer because you'll need to amplify the forward strand of DNA and the reverse strand. And then finally, we added water so that each sample could total out to 25 microliters. This value can vary based on what you're doing, but from my experience, it is pretty standard to have a PCR reaction that totals in the 25 microliter range. All right. Once we have our mixtures ready, we can go ahead and place them in a thermocycler. A thermocycler is this really fancy and precise machine that is able to raise and lower temperatures very rapidly. This is needed because PCR requires a variety of temperatures to replicate the DNA. What you'll do is set the program temperatures based on what nature needs and what your primers need. Using the image on the right as an example, this guy right over here, Step 1 shows the initiation of PCR, where the DNA in the reaction mixture is going through denaturation, which is effectively breaking the hydrogen bonds holding the two strands together and allowing them to become independent and individual strands. Step 2 is the completion of denaturation. Step 3 is where annealing occurs. Annealing is where the primer binds to the strands of DNA in your sample. Once the primer is bound, the temperature then rises again and induces elongation, which can be seen, there it is, in step 4. In this image, Taq polymerase will be rapidly adding nucleotides to the DNA strands that have primers bound to them. This allows target DNA to be elongated, while any remaining unwanted DNA will not be amplified. The PCR cycling program you set up will run for the desired number of cycles, constantly repeating steps 2 through 4, which is how we achieve amplification. Each cycle of steps 2 to 4 can take a few minutes, so running 25 to 40 cycles will be a lot of minutes. This is usually the time to go and be productive somewhere else, do other assignments, other experiments, what have you. When the program is finished running the DNA, is often stored. When the program is finished running the DNA is often stored at 4 degrees Celsius so that your amplified product doesn't denature. I know I mentioned earlier that you could make as much DNA as you wanted, which theoretically, if you have unlimited materials, you could make unlimited DNA. However, when running experiments, there is a conventional limit. Once you pass 45 cycles, it is more likely that non-targeted DNA will be amplified and could skew your results. Sorry. That's why the lower the cycles, the better, with about 20 cycles being optimal. This can vary, though, based on how much DNA is in your product and a few other factors. It primarily is just the focus of the DNA in your product, your PCR product. As you can see by the green arrows at the bottom of this program, these little guys down here, steps 2 through 4 will be cycled about 30 times. Well, not about, they will be cycled 30 times. This was done because we were working with lower concentrations of DNA, so we wanted to get as much amplification as possible while still being accurate. Now, with that whole mess out of the way, the PCR sample can then be visualized. In the case of my research, I'd run the PCR products through gel electrophoresis, producing a gel that can be seen as the image on the left. If the DNA is present, this image right here, if the DNA is present, then bands will appear at molecular weights denoted by a molecular weight ladder on the gel. And this guy right here, if you can see it all the way to the left, this is your molecular weight ladder, which lists each weight in base pairs. So each bar is like a different weight. So this could be like, as an example, like 500 base pairs and then 400, 300, etc. It varies based on what you're using. There's like hundreds, thousands probably of different molecular weight ladders you could use for your studies. This is because, these weight measurements are set up the way they are because larger strands of DNA will proceed through the gel more slowly than smaller ones. This means that larger strands tend to deposit towards the top, while smaller ones deposit towards the bottom. Any bands that are kind of in the center of the gel, so that would be these guys here, indicate that DNA was present in our sample. So we can see that since these deposited up here towards the top of the gel, they're probably a little heavier, a little bigger strands, and there was DNA in the sample. Since primers are so small, they will be the bands appearing at the bottom of the gel, often below your ladder. So these kind of faded guys right here, some of this might be DNA sample. It's a little hard to tell because it's very minimal, but generally there's usually pretty strong banding down here, and that's just where the primers that didn't bind or came off are going to deposit because they're so small. So the conclusions and next steps you gain from this will depend on the study that you're performing. All right, moving on to the next one. Reverse transcriptase is a special little enzyme that disrupts the central dogma. If you've never heard of the central dogma before and are instead thinking about central dogs, like I did when I first heard of the central dogma, you are not alone. The central dogma is a theory that DNA goes to RNA by transcription and RNA goes to protein by translation. Reverse transcriptase disrupts this because it allows RNA to go back to DNA. As is seen in the picture I've provided on this slide, the target RNA sequence gets bound by a primer. Here's our little picture. Here's our little primer. This primer allows reverse transcriptase to bind and sends RNA effectively back to DNA. What happens is reverse transcriptase turns all the uracils back into thymines. This is actually how RNA-based viruses are able to replicate in a host, since they do not carry any of the replication machinery themselves or even their own DNA for that matter. When RNA is reverse transcribed into DNA, we like to call this complement DNA, or cDNA for short. Oftentimes, you're going to see it listed, actually, like 99% of the times, you're going to see it listed as cDNA, because that's just the conventional standard for listing complement DNA. Performing reverse transcriptase PCR or RT-PCR is actually very similar to performing standard PCR. The biggest difference is what I mentioned on the previous slide. It just requires an extra step to get the RNA to DNA. Similar to PCR, you'll get your sample and isolate and purify the target RNA. This RNA will then be incubated with reverse transcriptase to form cDNA. From here, it is all the same steps as PCR. cDNA will be taken and put into a PCR reaction mixture. That mixture will be put into a thermocycler and run for 20 to 40 cycles based on your need, and then your DNA will be ready for visualization. Just had to get a sip of water there. Okay, figure 2a. After the longest detour ever, we finally made it back to the paper. Although hopefully on that detour, you were enjoying the journey more than looking forward to the destination, as it goes with most things. In this figure, the authors utilized RT-PCR to determine what the expression levels of each relevant GSDM were in each of the cell lines. PJVK, which we can see down here at the bottom, was their negative control, and GAPDH, all the way at the bottom, was their positive control. These controls are to ensure that the gel is working within its normal parameters of either being yes, there is something banding on this gel, or no, there isn't something banding on this gel. In this figure, they're showing that GSDME expression is high in HK1 and FADU cells, but is expressing low in C666-1 cells, which can be observed by the banding. So we can see we have our HK1 FADU cells. If we come down to GSDME, we see we got some fluorescent bands here. If we look to C666-1, it's like kind of faint, so there's minimal expression, but they aren't considering this significant. These two guys right here, they are considering significant. Although GSDMB, which we can see all the way up here, is expressing highly in all three cell lines and pretty pronounced, they did not attribute it with pyroptosis, so it was not included further in the study. Something to note is that RT-PCR was used because just isolating the DNA and running it does not ensure that you are amplifying the stretch of DNA that actually codes for these proteins. This is partly due to exons and introns and a whole host of other environmental factors that can impact what a stretch of DNA expresses. Using the mRNA means that it has already gone through alternative splicing and is heading over to the ribosomes, where it will be translated into a protein. The authors did not isolate the proteins themselves, as it is a longer task than RT-PCR. The product yields of protein isolation tend to be much less than RT-PCR, since the proteins cannot be amplified in quantity, only in the available amount currently in the cell can be visualized. So in other words, if they take the proteins, they can't then take the proteins and run it through a PCR and amplify it. All they get is the quantity that's just currently available in the cell at the time. So if there isn't a huge quantity, it can make it difficult to visualize or to provide evidence that something's going on. All right, figure 3a. In this figure, contrary to what I just said, the authors did perform a protein isolation. This differs from the previous figure in that the authors wanted to determine how much of a target protein was in the cell at that time. In this case, they were looking at the major players in the cell death pathway, which are BAX, BAD, BAC or BAC1, whatever you feel like saying, caspase-3, and the cleaved version of caspase-3, which they have just labeled as C-caspase-3. In the in the HK1 and FADU cell lines, we can see that down the TPL column, there is banding. So we look to our HK1, FADU cell lines. If we look to the right, TPL column, BAX, BAD, BAC1, caspase-3, C-caspase-3, all are banding, including in the FADU lines. This indicates that TPL is inducing the cell death pathway, because when we look at the blank column, which is to the left over here, where there is no TPL exposure, the cell death pathway remains inactivated. And the way you can tell is if you're just comparing the bands. So BAD isn't being expressed very highly in this, and it's not being expressed very highly here. CCAS-3, CCAS-3 here. These two are kind of important because when BAD merges with BAX to form the BAD-BAX complex, that's when they'll localize into the mitochondria. That's when Cas-3 gets cleaved, and then that's when the cell death pathway kind of starts kicking into gear and expressing all the other factors that are going to induce death in that cell. When we look at C616-1, we can see that there's little to no cell death pathway activation when exposed to TPL. Once again, they are also using GAPDH as their loading control to make sure the protein is transferring properly. Something else to note is that if you look in the blank column, C616 doesn't really have much activation of cell death in general, which is kind of what makes it such a potent carcinoma cell, because if it's not really expressing the cell death pathway, but it's having major issues, then you're just going to have a bunch of non-functioning cells roaming around, which is what makes cancer so detrimental in the first place. Okay, we're going to step away from the paper again and take a moment to talk about our next and final PCR technique, qPCR. The Q stands for quantitative. Effectively, you're turning PCR into a conclusion-based technique rather than a visual measurement that is usually just used as a step in the research process. The biggest change between qPCR and PCR is the addition of a fluorescent dye added to the PCR mix, which is called a master mix in qPCR, and a fluorometer to the thermocycler. This allows for real-time measurement of the DNA amplification as it's happening, and often giving qPCR the name of real-time PCR. Once again, the naming conventions of the scientific community have let us down. Why it's called real-time PCR and there's another PCR technique called RT-PCR, yet they aren't the same, is literally just confusion waiting to happen. Anyways, this is a real benefit, as you can watch the graph populate as the program runs, which I'll show you on the next slide. This little image that I have over here to the left isn't really the best example of like qPCR in a plate, but basically it involves making a ton of little tiny master mixes, and you just fill this whole 96 well plate with them. And I've only done it once, and it was a huge pain. It was really difficult. I mean, as you get better, it gets easier, but I mean, qPCR, it does take a lot of practice to get proficient with it. All right, so some of this is just repeat information from the last slide, but the biggest takeaway here is that qPCR allows for a very precise quantification of amplification of what's going on in your sample. Gene expression can actually be measured in this way by using RT-PCR and qPCR in the same go. You're just going to incubate your sample with reverse transcriptase, add some fluorescent dye, and then throw it into the thermocycler. Of course, it's never that easy, and there's some steps I'm omitting, but that's the gist of it. If you look over here to the left, you can see an output of a PCR reaction from a thermocycler that includes the, of course the word's escaping me, the fluorometer. There we go. On the x-axis, which is down here, it's kind of difficult to see, showing the normal of cycles, and on the y-axis, which is kind of impossible to see, it is showing the fluorescence. Yeah, it is showing the fluorescence. As your product increases, so too does the fluorescence. So it makes sense that over the course of more cycles, you'd have more fluorescence. So over time, or in this case over a course of cycles, fluorescence increases, and then you get all these bands that are heading upwards in the positive direction, and you can visualize amplification. And this does populate in real time, so you'll watch these data points start showing up as the reaction goes on. Okay, we've made it back to the paper, which now we're looking at figure 4b. And hopefully we are more equipped in knowledge, so hopefully we'll be able to understand what the authors are trying to show us with this figure. The authors used, for researchers, qPCR to measure the gene expression of c-Myc and hexokinase-II in the 3 cell lines. c-Myc promotes the expression of hexokinase-II, so without c-Myc, hexokinase-II cannot be expressed. They subjected each cell to a different dosage of TPL, which we can observe on the x-axis, which we'll see right down here. On the y-axis, they provide change in RNA expression, mRNA expression, right here. And usually for this, the reason why there's no measurement next to it is sometimes they'll just use arbitrary values, which just means values that are relevant to, their experiment, but don't necessarily have a unit associated with them. We can observe that each cell line showed significant decreases in mRNA expression of c-Myc and hexokinase-II when exposed to 150 nanomolar of TPL, which we can see right there, 150, 150. These stars are going to indicate significance, and in each of the cell lines, we can see that they have it. Over here to our left is going to be c-Myc, to our right is going to be hexokinase-II, which you can see denoted right here with the little boxes, which I know are a little difficult to see because the picture is copied over at like 180, p instead of being like 4K or whatever the paper was in PDF form. But if you need more clarity with these figures, you can just hop over to the paper and get a better look at them. So, if we reflect back to the first figure I showed you, this is a very similar story to what they found in their cell viability assay, which also showed 150 nanomolar was their magic number of TPL dosage as well, which was showing significant decreases in the viability of those cells to survive. Okay. Intermission, figure 4F. Now it's your turn to do some figuring out. Take some time, go and answer the intermission question on the reading guide, although it's basically like a late mission because the presentation is almost over. Okay, welcome back. I hope that figure didn't give you too much trouble. If you got stuck, that's alright. I'm going to provide a brief rundown of the figure. Figure 4F is showing us qPCR once again. This time, though, they're looking at compounds involved in metabolism. Here, they were trying to determine if their data in figure 4b can be corroborated. They show that upon exposure to TPL, each cell line shows significantly reduced glucose consumption, lactate production, and ATP formation. All of these are critical to the survival of the cell. Without them, a cell will die, which once again provides evidence to support the cell viability assay from figure 1a. Alright, here we have figure 9, the pathway, which finally we are nearing the end of this epic journey. Here, the authors most graciously, and with likely a lot of effort, provided us a molecular pathway roadmap for what they tested throughout the experiment. This figure can really help to put together everything that we've discussed today. I'm not going to spend much time on it since it goes a little beyond the scope of this presentation, but feel free to go over it, shoot me an email with any questions you might have. This is also a great tool for answering some of the reading guide questions, as well as understanding the paper as a whole. Okay, so on to the main findings. To finally conclude, the authors found that exposure to TPL reduced the viability of C666-1, HK1, and FADU cell lines to survive and function. They found that TPL caused the bad-Bax complex to translocate into the mitochondria. This induced a cell death pathway, causing caspase-3 to cleave. When cleaved caspase-3 became activated, it cleaved GSDME at its internal linker, causing GSDME-N, named so for the cytotoxic N-terminal, which forms the pores, to head to the cell membrane and create a pore. This caused the cell to die and gave us the term pyroptosis. Although I focused us more on GSDME, the authors also found that TPL can inhibit the expression of NRF2. NRF2 is important because it would normally inhibit arrestin. Arrestin itself inhibits the cysteine glutamate antiporter, which brings cysteine into the cell. Cysteine cannot enter the cell, it is not able to maintain the homeostatic balance the cell needs, and causes ROS to accumulate, leading to cell death, as mentioned in the introduction. So to say it in other words, without the expression of NRF2, the cell will undergo cell death. This form of cell death is called ferroptosis, which is more widely known than pyroptosis. Finally, that's the end. You've reached the end of the journey, and you are now free to enjoy your life once again.